Cell surface receptors, insulin, epinephrine, glucagon, these bind G protein coupled plasma membrane receptor on, and this extracellular signal is transduced to intracellular cyclic AMP, which function as a second messenger. And increase in rise in cyclic AMP or transacting factor, that is cyclic AMP response element binding protein CREB protein, is phosphorylated and activated by protein kinase A. And this active CREB protein binds via a leucine zipper motif to a cis-acting regulatory element, that is CRA, that is cyclic AMP regulatory element, and that results in transcription of target genes. Example, the phosphoenol pyruvate kinase enzyme, glucose 6 phosphatase enzyme. Here, cyclic AMP response element, cyclic AMP stimulates protein kinase A, CREB binds, and phosphorylation of CREB takes place, and transcription is activated. Now the regulation through variation in DNA, access to DNA. Active chromatin contains histone, and reversible methylation, acetylation or phosphorylation by histone acetyl transferase and histone deacetylase takes place. But these modifications decrease the positive charge of these basic proteins and decrease the association with negatively charged DNA. And this relaxes the nucleosome and creates the site, and the transcription factors can access to the specific regions. The transcriptional genes are less methylated. That shows hypermethylation silences gene expression. The amount of DNA, an increase in number of, that is known as gene amplification, takes place in normally in non-mammalian, but in mammalian it is seen with some diseases and in response to particular chemotherapeutic drugs. Example, methotrexate, which is an inhibitor of enzyme dihydrofolate reductase, which is required for the synthesis of thymidine triphosphate and which is essential for DNA synthesis. But what happens when patient takes methotrexate, gene amplification takes place and the cells, malignant cells, results in an increase in the number of DHFR genes and resistance to the drug and allowing TTP to be met. Arrangement of DNA. Here is the permanent rearrangement of DNA takes place during immunoglobulins production by B lymphocytes. Single variable B diversity D joining J genes brought together to form unique variable regions in immunoglobulins. And that leads to generation of 10 to the power 9 to the 10 to the power 11 different immunoglobulins from a single gene, and thus provide recognition of an enormous number of antigens. The pathological DNA rearrangement are seen between chromosomes when the translocation takes place. Mobile DNA elements. Transposons are mobile segments of DNA and their movement is mediated by enzyme transposase, and they are encoded by the DNA itself. When they are direct, then cut and paste. When they are in a replicative manner, then copy and paste. And there are certain replicative transposition made by reverse transcriptase. Then it is known as retrotransponon, and which alter the gene expression and causing diseases such as hemophilia A and Duchenne muscular dystrophy. And the growing problem of antibiotic resistant bacteria is due to these mobile DNA elements. Here we will discuss about messenger RNA processing. You see messenger RNA capping and polyadenyl tail Messenger RNA processing. Capping at the 5' end, polyadenylation at the 3' end, and splicing are the essential for the production of a functional eukaryotic messenger RNA from most pre-messenger RNA. And these variation in splicing and polyadenylation affect the stability and gene expression of the messenger RNA. Alternative splicing takes place. Pre-messenger RNA for tropomyosin undergoes tissue-specific alternative splicing to yield a number of isoforms. Over 90% of the human genes undergo alternative splicing. Here is the gene and here is the takes place alternative splicing and produces protein isoform 1 and protein isoform 2, and both are having different function. Alternative polyadenylation. Some pre-messenger RNA transcripts have more than one site for cleavage and polyadenylation. And these alternative polyadenylation generates messenger RNA with different 3' ends and thus altering untranslated region or coding sequences. And this APA is involved in the production of membrane-bound and secreted form of IgM, this alternative polyadenylation. Messenger RNA editing. This is post-transcriptional modification takes place. Example is Apolipoprotein B. Here in intestine, it is synthesized in intestine and liver both. In what happens in intestine, cytosine base in CAA codon is enzymatically deaminated to uracil and thus convert into a stop codon, that is nonsense codon, and result in a shorter protein, that is ApoB48, that is only 48% translation of the gene, and it is incorporated in the chylomicron. But in liver, 100% translation and ApoB100, which is the apo protein of VLDL. Messenger RNA stability. They are very stable, but duration in cytosol before degradation influences how much protein product can be produced. These messenger RNA exist as ribonucleoprotein particles in cytoplasm, and they sometimes protect or sometimes promote the nuclease enzyme attack on the messenger RNA. Certain effectors such as hormones regulate the stability of messenger RNA by increasing or decreasing the number of these RNPs. The 5' structure prohibits the action of 3' exonuclease. The deletion of 5' UTR results in the threefold prolongation of the half-life of c-Myc messenger RNA. Shortening of coding region prolong the life. Absence of poly-A tail, rapid degradation of messenger RNA. If there are AU-rich regions, then have a very short half-life. Example, encoding oncogenes or proteins and cytokines. Here is the example of regulation of iron metabolism and gene silencing process of RNA interference. Regulation of iron metabolism. Transferrin, a plasma protein, transports iron. It binds to a cell surface receptor, that is transferrin receptors, get internalized, provide cells with iron. The messenger RNA for TFR has several cis-acting iron response elements, that is IRE in its 3' UTR, and IRE binds to transacting iron regulatory proteins. When iron concentration is low in the cell, IRFP binds to IRE and stabilizes messenger RNA, allowing transferrin receptor synthesis. When iron concentration is high in the level, IRP dissociate, and that leads to messenger